Our project is titled Exploring the Impacts of Genetic Modification on the Preservation of Endangered Species. Due to many environmental factors, we are seeing a decline in many species populations and destruction of their habitats. Deforestation and unsustainable agriculture are two that are driven by the way us humans manufacture food today. Fertilizers and pesticides cause erosion, runoff, and the contamination of water sources. Overfarming and overproduction of plants can drain the nutrients and life out of land. And GMOs can cause plants to be resistant to certain parasites and diseases. In addition, there is a great amount of illegal wildlife trade with the poaching of elephants and tigers. Many of these species, especially keystone species, which are species that many other organisms in a habitat depend on for function of the ecosystem, are close to extinction. These species prevent overpopulation of prey, provide food and shelter, or can regulate microbial activities. When a population experiences a drastic decrease in number, it's called a population bottleneck. This occurs when the allele frequencies in the surviving population differ from those of the original population after a great environmental disaster. While the reduction in population number is temporary, the population will recover, but with greatly reduced genetic diversity. The new population contains mostly one allele for the gene, as we can see here is the red allele. This can be detrimental for the future of the population because they are so genetically similar, and they can potentially be easily wiped out by another disease or environmental disaster. Biodiversity is extremely important to have on our planet, especially for these 5 reasons. It is important for our economy and jobs, ecological life support and food security, recreation and tourism, culture, art, aesthetics, and physical and mental well-being, and scientific research. One way genetics can resolve this issue is through manipulating genes and thus controlling the diversity of species. This figure shows us the basic idea of DNA knockout technology and a strategy for creating a knockout mouse. DNA knockout technology works by generating a null mutation in a target gene that is then introduced into an organism using transgenic techniques, usually through embryonic cells. The blastocyst that is formed is placed into the uterus of a surrogate mother mouse, sometimes called a pseudopregnant mouse. The mother gives birth to chimeras, which has both the knockout genes and the genes of the blastocyst, and cause a loss of function in the targeted gene. The chimeric mice can then breed with black mice to produce heterozygous children, and these heterozygous mice can then interbreed to form homozygous mice for the knockout genes. Similar technology can be used to insert target genes into organisms with using a target vector that would contain a copy of the gene of interest that has been mutated and inserted into a large segment of foreign DNA.
Genetic modification is not a new scientific idea, although its long-term effects on endangered species at this point in time are relatively unknown. Some research has been conducted on endangered species who face serious threats to extinction. The Frozen Zoo is located in San Diego and contains cells from hundreds of species. The population numbers of the drill and the northern white rhinoceros are extremely low due to the actions of humans. Scientists were able to successfully reprogram adult fibroblast cells into induced pluripotent stem cells. The results of their work included the production of germ cells, which have the potential to be used in future reproduction efforts. This would allow for increased genetic diversity in low population species. On December 10, 2020, a black-footed ferret was cloned from an individual who had died approximately 30 years prior. While this endangered species suffers from low genetic diversity, they also suffer from diseases such as the plague. The successful cloning sparked curiosity in scientists who started attempting to insert an anti-plague antibody into the DNA of mice. Because endangered species have low population numbers, it is risky to experiment on individuals of the population, so it is common for scientists to begin testing with lab mice. Other successful results regarding genetic modification can be translated over to endangered species. For instance, the creation of hypoallergenic cats using CRISPR gene editing shows the possibility of editing unwanted traits out of species. Genetic modification in endangered species has produced successful outcomes on individuals and in cell research, but when these individuals are reintroduced to their wild populations, serious consequences can arise that need to be considered.
There are also plenty of benefits of genetic modification. Transgenic mice are used in labs and can be studied for basic learning biological functions that can be translated to humans to gain a better understanding of diseases and even have the potential to develop cures. There's also xenotransplantation, which is when animal organs are genetically modified and used to be implanted into humans. This can combat long transplant lists that are becoming a growing problem with extended lifespans and many patients not being able to get the organs that they need in order to survive. There's also the improvement of breeding and food quality, as well as the development of pharmaceuticals from these transgenic mice. An example of a pharmaceutical is insulin that was developed from the pig pancreas and is now used in humans. Some benefits specific to endangered species is that it can increase the biodiversity in an ecosystem like it was previously mentioned. There's also the preservation in which we can improve their understanding of microevolution through the better understanding of how these species have been able to survive all this time. We can lastly use the use of genetics to modify species and increase the reproduction rate and decrease the possibility of extinction in these endangered species.
All right, so now there are some potential consequences that may result from gene modification in general that may be applied to genetically modifying endangered species. Now, these consequences on this slide involve the modification impact on the general ecosystem, which may disrupt its balance. First, we have horizontal gene transfer, where the recombinant genes may intertwine with that of another species. Now, for example, this could be like passing on an antibiotic trait to pathogens, which would allow them to thrive in harsh environments, which would be detrimental to the ecosystem itself. Next, we have competition with natural species. In this case, the genetically modified organism, endangered species themselves, would become invasive and overwhelm the majority population, leading to potential damage that may not be restored. This ties in with the next point involving the difficulty to follow the gene. As the species begins to breed, the gene may be lost in the population. So if a problem were to arise, it would be hard to track and difficult to track down the gene that caused it. Finally, interbreeding may present a problem depending on how favorable the genetically modified traits are compared to the natural one. If it is presented as a less favorable trait, the gene will begin to disappear in the following generations to come.
When discussing the application of genetic modification in endangered species, it is necessary for us to consider a range of negative impacts associated with this practice. Animal welfare comprises a large portion of the ethical concerns associated with genetic engineering. Due to the fact that many of the embryos that undergo genetic engineering procedures do not survive, and those that do survive, only a small proportion carry the genetic alteration of interest, a large quantity of animals is required when attempting to genetically modify a population. This poses difficulty when attempting to modify an endangered species, as their population count is likely to be quite low initially. Animal welfare concerns can be further addressed using the 3 R's: reduction of animal numbers, refinement of practices and husbandry to minimize pain and distress, and replacement of animals with non-animal alternatives wherever possible. When altering an organism's DNA, it is important to bear in mind that this alteration may disrupt that organism's genetic equilibrium in unpredictable ways and may result in negative changes to the subject's behavior and well-being. In this image, we have a transgenic pig. This pig had its growth hormone modified, and this can result in arthritis, stomach ulcers, heart hypertrophy, liver and kidney disease, infertility, as well as other conditions when growth hormone overexpression occurs.
To tie in this topic that we've found up ourselves, endangered species may be provided an avenue to avoid anthropogenic extinction and genetic redundancy due to reverse research approaches. We must also consider benefits that can apply to these endangered animal populations in their natural habitats with CRISPR and other genetic modification techniques. Lastly, though, we must keep in mind costs and potential consequences to these individual organisms and their populations with unforeseen effects and other ethical concerns. Lastly, these are our discussion questions, and we will go over these in a later time. Thank you so much. These are our references.